one was point mutation, in which any one of this, like any one of these nucleotides, if they change their position, how from glutamic to valine, it can change the normal RBC to a uh, sickle cell anemia or sickle cell RBC, which is a defect and causes severe problems. So that is point mutation. Now then we have even started with frame shift mutation. Frame shift mutation means complete shifting of either because of the addition of more than one nitrogenous base, or removal of, I mean, either addition of more than one nitrogenous base or deletion of more than one nitrogenous bases can make a lot of changes, physiological changes in the body, and that is because of the complete shift of the frame, complete shift of the changes in the I mean the nuclear or the what you call it nitrogenous bases, which uh, completely is unreadable or uncodable properly. And that is what we learned with one statement they were trying to explain, like the cat ate the rat was the statement. With that they were trying to explain about uh, point mutation further with the frame shift mutation. uh, Frame shift mutation we have to see, so we'll
Either
proceed.
inserted or deleted from the DNA template. For instance, in our
Okay.
example, if we insert two letters R and K, it will change the reading frame, and hence the altered statement will read, "The cat arced at her at." Whereas, if we delete the two letters, that is one C and one A, from the original statement, the altered statement will read, "The
Okay. uh, the cat, you can see this, what they're showing you was, first example was in that was arc, that one, huh? The arc, the first one, in which two genes were substituted. Okay. In that the statement completely changed. Instead of uh, the cat ate the rat, the statement changed. It was not a normal statement, the arc, something like that, it was coming up. Now here, that is one. Here they are showing the deletion. When two, instead of substitution, if there's a deletion, two getting deleted, what will happen when two gets deleted? Where it added was, see, from where it went, uh, the is still right, fine. After that it was cat cat, then it was ate, okay, then then rat. Okay, 1 minute. Okay, so okay, I wanted to show you with a pen. When where the from where the point from where the deletion happened was, from where the deletion happened was from here, okay, here, till the, and sorry, right from here, it was, the statement was the, there is no change. Okay, then it was cat, cat. Okay, in this cat what happened, two letters a, two letters C and A got deleted. Okay, when C and A got deleted, what happened, this T took the, it wants two more codes, so it automatically, it took the next from ATE8, it took two other nitrogenous bases, so the word became TAT. Okay, right, then the word became TAT. And again, when it took it from 8, the E was left here, so E took it from the, THE, and this the came here, you see. and further rat here. But when they readjusted themselves, T1T is single there. Okay, you can see when the deletion happened for more than one gene, it has this nucleotide or nitrogenous basis adjusted itself in this way, which has really no meaning at all. If the sentence is not giving you a meaning, that means, this is an example, same way the codes for nitrogenous bases, which codes for proteins and all, will give you no meanings, no proteins will be synthesized, or the proteins which will be synthesized will be of no use in our body. That is why a lot of problems, yeah, physiological changes and problems are seen in a person where mutation happens for more than one genes, more than one gene. Is this is understood? With this statement they are trying to explain. Okay, the correct statement was the correct combination. They are saying like triplets. We learned yesterday in the genetic code. In the genetic code we have learned yesterday that three nitrogenous bases together forms one, for one code for one amino acid. And in that way we have learned, uh, if if one gene changes or mutation happens, what can be the things. While when deletion happens, that is, this is frame shift, this is complete frame shift. How you understood? No, from here THE, when THE, there is no change, no mutation happens, so it is intact, this one. Okay, the problem came from this area, है ना? From where the CAT, when CAT, what happened? CAT changed, so it took C and A has deleted. So what it has done, it has taken the other, when C and T has been deleted, what it did, it took T and the other letter from the next amino acids nitrogenous bases. So this, though even the statements, you can see it is completely, it has shifted from one to other end, right? Did you understand, children? This is how great difference happens. And that's why like in phenylalanine, we have learned that when the one of the disorders, when we learn Klinefelter's, okay, and we have learned about some disorders, Down syndrome, Turner syndrome. In this, lot of changes in the organism, you can, in the human being, you can see, because it is complete chromosomal mutation takes place, a whole increase or decrease of chromosome takes place, the whole. When we say chromosome, it is a lot of sets of so many genes are present inside. So it makes totally a change in the physiology of the organism, individual, including the mental retardedness is also seen. So if this is understood, this with this statement, did you understand what they mean to explain you,
Yes,
children?
ma'am.
Yes, ma'am.
So how even one or two letters in a word, if it is deleted and if it takes it automatically from the other, the statement is changing. This is an example. They're saying exactly like this, it happens inside the human body, inside the genes. If one nitrogenous or two nitrogenous bases gets deleted, they will adjust it like this, which will have no meaning and will not be, a person will not be no more normal. Okay, we'll proceed. So
Cat eth era T. Likewise, in a frameshift mutation, if three or multiples of three bases are inserted are deleted, it will result in the deletion or insertion of a codon. This in turn will result in the creation or deletion of an amino acid. Interestingly, frame-shift insertion or deletion of one or more bases changes the reading frame only from the point of insertion or deletion. This point is clear when we compare the original statement with the statements that represent the mutation. Did you know that frame-shift mutation is common among textile workers? This is because they are often exposed to acridine dyes such as 5-aminoacridine, which enters their bodies through inhalation or physical contact. These dyes get intercalated or wedged between two adjacent purines and thus increase the distance between them from 3.4 angstroms to 6.8 angstroms, leading to frame-shift mutation. Chemical, physical, and environmental factors thus lead to the addition or deletion or replacement of one or more base pairs of a codon, which in turn causes frame-shift mutations and point mutations. A series of experiments by scientists and biochemical experts proved that both RNA and DNA served as genetic material. However, the question
Okay, I think it paused. Yeah, actually, what we just saw, the acrylamide dye was actually an occupational disorder, okay, disease rather, you can say, in which the mutation is happening. You might have heard occupational diseases. There are some diseases which happens because of your workplace, okay, the kind of work you do, okay, these kind of the people who work in the textile industries and they work with the fine fibers, okay, what happens, this acrylamines are another kind, it's a chemical which which can this kind of things help in the mutation. And once this mutation happens, if you see this animation you see again at home, I mean during when you sit and study, kindly see how this chemical, when it what actually happens is when this chemical infects, or when we inhale these the workers who are working there in this industry, they inhale such things and it will enter into their genes, and that will create this acrylamines. They will create a distance between the genes, okay, and once they come and they actually interference into the the positions in the nitrogenous bases, and because of its presence, what happened, the distance between two nitrogenous bases increases, and that will make again a person little diseased. So there are some reasons. Some can be environmental reasons, natural reasons, sometimes no reasons for the mutation to happen, and mutation causes either point or frame, depending upon what kind of mutation happened. You can see the disorder of that level. And
question that arose was, what exactly made both DNA and RNA suitable genetic materials? The answer lies in the fact that for a molecule to
we
be
are
a
coming
genetic
back to
material,
first
it
topic.
must fulfill 4 simple conditions. It should be structurally and chemically stable, which means it should not change with age, change in physiology, or life cycle. Yet, despite stability, the molecule should provide scope for slow changes or mutation that is necessary for evolution. Further, the molecule should be able to express itself in Mendelian characters and also be able to generate its own replica to become a genetic material. Most
So these 4 characters we have learned already in the beginning as we started the chapter. I told you when during the search for genetic material, I told you that genetic anything to be any molecule, anything to be called as a genetic material, it should have minimum this four uh, important, very important characteristic feature. One of it is it should be stable. You understood? No, stable means physically as well as chemically or structurally. You can say very much stable. As you grow in age, the DNA should not change. Okay, the genetic material should not change. Otherwise, what will happen? The information what is carrying will not be valid. It will change. Okay, so the person will no more remain normal. So it is needed that the genetic material should be stable throughout. It number two, it should be allowed little bit very the genetic material should allow mutation. Okay, because mutation causes evolution, which is very important for the life to go on with the changes happening around the environment. If there is no change happening with the change in the environment, what will happen? The organism will perish because the survival of the fittest and those theories, you know it. Okay, then the third thing is or the DNA, or sorry, the genetic material must be able to replicate itself. Replication means making its own copy. Okay, and since they have this ability of replication, and the last quality or the last characteristic feature to be called as a genetic material is to be able to express itself in Mendelian characters. Mendelian characters means all those theories what you have learned are Mendelian characters. Basically, genes are expressed in all those theories what you have learned. That comes under Mendelian characters. So that all is very much satisfied by this DNA and RNA.
Molecules, including proteins, fail to fulfill these criteria. Even so, both the nucleic acids, DNA and RNA, meet all these requirements. Although it is to be noted that DNA serves predominantly as genetic material, RNA also acts as genetic material in few microbes such as tobacco mosaic viruses and QB bacteriophages, and also performs the role of a messenger and adapter. Of the two nucleic acids, it was DNA that played a part in Frederick Griffith's experiment.
What happened? I didn't pause it.
No, actually, I paused it. I was moving it.
And this was
I was
ahead, na?
bringing the topic. I was bringing the topic which we have to see actually.
Okay, but still little more things you have to learn the structure of that. They're not going to teach you how tRNA is formed, because we have learned how many types of RNA polymerase enzymes are required for the synthesis of or the making of the when I mean during the learning, when we learned about how messenger RNA is formed, we learned about RNA polymerase enzymes. Now the various types of polymerase enzymes differs in eukaryotic cells, which helps it in making of tRNA, various units of rRNA and hnRNA, which later on after some process it becomes mRNA, right? So all this we have learned. So we are not going to learn the process of making of tRNA, but yes, this tRNA has little story behind. Like it was first called as adapter molecule. It was an hypothesis given by one scientist. That is, there's one during for the process of uh, protein synthesis. It was first an hypothesis that uh, protein synthesis other than the ribosomal RNA as well as the messenger RNA, there is one adapter molecule. You understand what is adapter? We use at home electronic device. It is like adapter. So same way, tRNA was first given the name as adapter molecule. That's what is still known as adapter or tRNA, okay, transfer RNA, since it helps in you know taking the nitrogenous base. It brings the nitrogenous bases along with it, and with the help of an enzyme and its structure, everything we're going to learn. Then we will learn translation. Okay, so we'll move with the next
Human
topic.
cell is an amazing structure which is busy carrying out various life-sustaining processes, one of which is protein synthesis. For this process, it is essential that amino acids which lie scattered in the cytoplasm are carried to the messenger RNA or the mRNA template, the site of protein synthesis. Till the 1950s, scientists did not know enough about how proteins were carried to the mRNA. The breakthrough came in 1955 when Francis Crick, who had earlier revealed the DNA structure and its properties, postulated the adapter hypothesis theory, in which he stated that it is therefore a natural hypothesis that the amino acid is carried to the template by an adapter molecule, and that the adapter is the part which actually fits onto the RNA. In its simplest form, this hypothesis would require 20 adapters, one for each amino acid. Soon after Crick published his hypothesis, scientists across the world worked to find this mysterious adapter molecule that adapts itself to carrying amino acid and reading it to the codon of mRNA. Success, however, came to Robert Hawley when in 1965 he discovered transfer RNA or tRNA, the adapter molecule, and also worked out its entire chemical structure. Today we know that tRNA, the smallest of all RNA molecules, is made up of about 75 nucleotides. Synthesized from a DNA molecule in a process similar to the synthesis of mRNA, tRNA is soluble in 1 molar NaCl and is hence known as soluble RNA or sRNA. Each tRNA has G at its 5' end and CCA at its 3' end. Interestingly, tRNA takes the shape of a clover leaf when examined as a two-dimensional structure, whereas it looks like an inverted L-shaped molecule in a three-dimensional structure. Regardless of its dimensions, a tRNA consists of 4 arms or sites. Ribosome.
Okay, so a small history understood about that Robert is the one who has given this chemical structure what you see right now, known as the smallest one, the smallest among the all mRNA, but comprises of how many nucleotides, nitrogenous bases? 75. Okay, since it is soluble in 10 molar, it is known as soluble RNA also. Now this is the story about adapter molecule, or which is known as tRNA. We are going to learn about its structure. Now, this structure is important to learn. Again, very important diagram this is, and you have to understand the each arm of it. Okay, they're going to explain you the arms. Each, as you can see, 4 arms around. All the 4 arms has a purpose, which and as you can see the codes along there, these codes are meant to attach itself somewhere. Okay, and for 20 amino acids there are 20 adapter molecules. Okay, each one very particular because each one has to identify the perfect part of the mRNA to fix itself to mRNA, to ribosomal RNA, as well as to bring a amino group. It has to, it needs to have all the 4 arms, a code, and it is particular for one. That's why there are 20 tRNA for 20 amino acids. Okay.
Recognition site, anticodon site, enzyme recognition site, and amino acid binding site or carrier end. Each of the tRNA regions perform a particular function. Take the case of the ribosome.
Okay. Now pen again has gone. Okay. Where the pen has gone. Mm, fine. Here, this is the side. This you can see, no, this side. This one, this site is known as a ribosomal recognition site. Okay, when it comes on the, like when mRNA is prepared inside the nucleus and it comes out of the nucleus in the cytoplasm. Meantime, what is happening is the adapter molecule I mean, from the cytoplasm. This is the area. This is the site. It for amine group to be, to be, yes, fine. Amino group to be, identifying site. This is enzyme recognition site. This one, that this site is needed for, it is this two. This is the first. This is the second, I will say. Okay, this enzyme recognition site, second site, the enzyme recognition site. What it does is, before it actually comes and like this is the fourth one, is the site which recognizes the uh, codes on the mRNA. This is like this code, no, it is written in the green color. This is the site which will recognize the mRNA, and where the like if it is written C, definitely you know C G C, right? So C means in the mRNA what should be there? G should be there, right? If it is written G, it should be C. So like that, we know that if it would have been A, it would have been made a complementary pairing with A with not T, U. Okay, so U, like that. So this codes, this one, this green color site is the site, or this site, the bottom part of this tRNA, this arm recognizes the mRNA and it fixes it there. This is happening. The last thing, what happens first is, it will, the first thing is, before, by the time mRNA comes on in the cytoplasm, what adapter molecule, what they do is, with the help of recognition site of enzyme, this one, an enzyme is present in the cytoplasm, which is very important to fix. So it will select the enzyme will attach here in this pink part, and from here, with the help of this enzyme recognition site and the enzyme from the cytoplasm, it will take one amino group here in the site 1. Okay, it will attach one amine group, and now it is activated or charged TNA with the enzyme attached and taking the uh, amino group. Now, after taking the amino group, only it becomes activated or active, or charged TNA. And then, as it with this, when amino group, it comes and sits on the mRNA, and where it will sit, that will be recognized by this green color site, this one. Okay, this site will recognize its which site has to attach itself, where it will attach to the mRNA. And then, while setting this, it has to also see that the two ribosomes which are present already on the mRNA, uh, two ribosomal parts will come and set itself. To recognize that site is this one, the purple one. It is the ribosomal recognition site. So this all four arms has got a very particular code, and these codes recognizes, like each keys recognizes a lock. Same way, each particular tRNA is having a definite, this, and after the work is done, it comes out of the mRNA, and again it is ready for the next translation. Okay, have you understood these four parts? Again, I will say, this is enzyme recognition site. This is enzyme recognition site. This is messenger RNA recognition or mRNA recognition site. This is ribosomal recognition site, and this is the site which will be taking up an amino group, selecting an amino group and bringing along with it. Fine. I think time is getting over.
Recognition